Assalamualaikum and hi, my name is Nurul binti Abdulaziz, S64348, and today we are from Group 3. I will discuss about the introduction for first lab, which is parasite. So fish as vital components of aquatic ecosystem and a source of nutrition for both aquatic and terrestrial organisms faces continual threats from factors like pollution and parasites. Parasitic infection in fish can lead to various adverse effects such as reduced growth and weakened immune system. There are two objectives, which are to compare the types of parasites found in each type of fish. Second is to identify parasites present in marine, freshwater, or canned sardines, which will shed light on the potential risks associated with the consumption of fish from various resources.
So next, I will be explaining about the materials and methods used in this lab. The materials used are sharp needles, glass slide, cover slip, saline water, compound microscope, scalpel, small scissors scalpel, forceps, freshwater fish, which is climbing perch, and marine fish, which is the penny stretching bream. So for the method, we will be using diagnosis method first, where the freshwater fish and marine fish were observed for any clinical signs such as lethargy, isolation, gasping for air, and poor body condition. And then we'll be using AQU3814 method, where the freshwater fish eyes were covered with damp tissue, and then sharp needle was put at above of the fish head where both of the eyes meet. This technique can instantly kill the fish without making it stress and therefore avoid the production of ATP. And then the damp tissue was uncovered from the fish eye after the AQU3814 method finished. And then the scraping of the skin method, where the part of alongside of lateral and ventral line near the dorsal pelvic and anus fins is scraped by using glass light at 45 degree. And then mucus was collected on the glass light. And after that, 1 drop of saline water was put on top of the mucus. Cover slip was solely placed on top of the mixture and mucus, and the sample was then observed under the microscope. For marine fish, step 7 until 13 was repeated to be observed under microscope. For scraping of the gill method, the operculum of the fish was cut to obtain the gill rector by using small scissors scalpel, and then gill rector was cut into small pieces and placed on glass slide by using forceps. And then it was observed under the compound microscope. And next is observing the sardine fish, and then all fish were taken out to be observed. The fish flesh was scraped and put on top of the glass slide. The sample was observed under microscope.
Okay, my name is Najwa, S62913. For the result part, after the diagnosis process was carried out on these two types of fish, we found trematodes in the marine fish sample, which is Nematodes species, at the skin area. For freshwater fish, which is Anabas species, nematodes are found in the gill area. And for the processed product, which is sardine, there is no parasite have been found.
Now, I will continue with discussion. The parasite that was found in our marine fish sample resembled a Digyna trematode species because of the presence of marked sucker and random sucker seen in the slide. Digyna trematode can cause grub due to the node color appear on the fish skin, and if many nodules appear in the fish skin, it can become hypoxic and can also be fatal. Based on our fish, no nodule seems to appear on the fish skin. This may indicate that the Digyna species was not in the infectious stage, or it's still in the early stage of infection.
We move to the discussion. During the observation, we found a nematode species on freshwater fish, which is it inhabit in gill of the. So, do you ever know about fungus staining technique? So here I will talk about fungus staining technique. It can be broadly categorized into. So first is wet mode preparation, and the second is differential staining. A wet mode is a fungal staining technique performed by suspending the specimen in the reagent solution and observe it in the microscope. LPCB or lactopanel cotton blue commonly used for mounting food for preparation of microscopic slide. The panel acts as the disinfectant and kill the fungus. The lactic acid, it preserves the fungal morphology. For glycerol, it prevents the sample from drying, and the cotton blue, it stains the outer wall of the fungus. So the objective for this study is to studying the morphological characteristic of fungus that help in detecting and identifying the fungus.
Hi, my name is Nurul binti Abdulaziz, S64348. For materials and methods, we need 100 ml of distilled water, cotton blue, dropper, slides, and fungal colony. So for the methods, the first method is we will coat a clean glass slide with a lactophenol cotton blue, and next we collected the fungal colony using forceps. The third one is the mycelium was evenly distributed over the LPCB. We will ensure that there are no bubbles present, and last step was examine it under 40 times of objective lens.
For the result of fungal staining, on the left side is the result for magnification 4 times, and the position is at the center of the fungus. As you can see, and the right side is the magnification 10 times, and the position is at the center of the fungus. On the left side is magnification of 10 times, while the position is at the center of fungus, while on the right side is magnification 40 times, and the position is also at the center of fungus.
Based on the lab experiment, there are two different parts of fungus that had been observed, which are the center part and the side part. The center part had the characteristic of pink color, its small structure which can be sampled by using forceps. Meanwhile, the side part had the micromorphology of powdery and fluffy white colored threads. It is a true fungus as the hyphae contain wall between cells that known as septa. True fungi normally found on expired bread as mold and known as eumycota, which is true fungus. True fungi are related more to animals and originated from opisthophons, meanwhile false fungi is related to plants and originated from straminophales. Example of fish disease by true fungus is aspergillosis, which is under the phylum of Ascomycota and caused by Aspergillus spp.
We're moving to micromorphology structure. At the first picture, which is 4 times magnification, we can see a general overview of the fungal sample. It helps in locating the area of interest and observing the overall structure and distribution of fungal elements. The second one is 10 times magnification, allows for more detailed observation, and the individual spores hyphae and more structure are more clearly. It is suitable for getting an initial understanding of the fungal morphology. And the last one is 40 times magnification. We can see a finer details of the fungal structure, and it is useful for closely examining the individual spores, the arrangement of hyphae, and any other intricate features. At this three picture, we can see a blue color occur at the fungal area. The fungal color is actually caused by lactophenol cotton blue solution that we usually use for microscopic observation of fungi. Among the function of this solution is to kill any living microorganisms, preserve the fungal structure, and stains the fungal stains wall.
We are able to identify that the sample was a true fungus as it has a smooth pink center with a powdery cottony outer layer which indicate that it was a true fungus. Other than that, with closer inspection, we are able to see the finer detail of the fungal structures such as spore, hyphae and other structure more clearly, that which help us indicate that it was a true fungus. And that's all from us. Thank you.
Hi, we are from Group 3. In this video, we will present about Lab 3 Practical Report, which is Nervous Necrosis Virus or NNV. Our group consists of 6 members, which is Estefania, Soresni, Elisha, Nuri Sofia, Eltasha, and me, Najwa.
First, what is NNV? NNV belongs to the beta nodavirus genus and is responsible for causing viral nervous necrosis (VNN) in fish. This viral infection primarily target the nervous system, leading to severe neurological symptoms and high mortality rates in affected fish. For the sign and impact on fish health, fish infected with NNV may exhibit abnormal swimming behavior, including erratic movement, swimming in circle, and a loss of equilibrium. Additionally, NNV can cause damage to the brain and spinal cord, leading to a range of neurological disorders. This virus poses a significant threat to both farm and wild fish population, affecting various species.
First of all, the first objective of this lab is to study the presence of nervous necrosis virus (NNV) in a fish sample using the NNV test kit. Second is to see how biosecurity measure for aquaculture facilities minimize the risk of nervous necrosis virus introduction and spread. And the last one, to investigate the interaction between NNV and its host organisms.
Next, we move to materials for this lab. First, we need an NV test kit, petri dish, disposable grinder, microcentrifuge, scissor, and dropper. Okay, so for this lab method, first step, we will take the right eye stalk from the fish using scissor, and then we will grind the eye stalk in petri dish. Next, we will take the tissue and insert it in the microcentrifuge. Then we grind it until mixed well. After that, we take the upper layer of the solution. Put 5 to 6 drops into viral nervous necrosis test kit. wait for 15 minutes for the outcome, either positive or negative. And in this lab, we got negative result for viral nervous necrosis.
So for nervous necrosis virus disease, we use areolate grouper as our sample, and then we use its organ, which is eyes. So according to the result on the test kit, it has no T line. Thus, it is negative for the NNV disease.
We move on to the discussion. So, what is VNN or viral nervous necrosis? VNN actually belongs to the beta-nodovirus genus of the Nodaviridae family. It is considered to be a serious disease of several marine fish species. If the test kit produced a positive result, this suggested that the fish was infected by the VNN infection. But based on our result, the test kit showed a negative result. NPV histopathology signs usually exhibit a vacuolation in the cell of the spinal cord, brain, and retina. As stated by Mandi et al. 2002, that the nervous cell usually show the most necrosis, especially in the larval and juvenile stages. Based on our fish sample, from our fish physical assessment of our fish retina, no necrosis indication was found.
In conclusion, our group has studied that there is no sign of NNV, nervous necrosis virus, in fish sample after using the NNV test kit. Our group also has observed the importance of biosecurity measures for aquaculture facilities in minimizing the risk of nervous necrosis virus introduction and spread, which one of the measures is by maintaining good water quality and hygiene to prevent the accumulation of pathogens and toxins. Our group also has investigated the interactions between NNV and the host organism, which is usually marine fish, where the virus will bind to specific receptors or co-receptors on the host surface cell as grouper optic nerve proteins and go into the cells through endocytosis. My name is Estefania Lourdes Anasima, S64498, and today our group 3 will present about white spot syndrome virus, WSSV. For my part, I will talk about introduction and objective of this lab. WSSV is stand for white spot syndrome virus. That is a virus that causes high economic loss in shrimp farming industry by trigger aerobic lipolysis in shrimp hepatocytes, which is first detected in Taiwan, then exposed to all Asian industries. WSSV is from the genus of virus and cause clinical sign of lethargy, appearance of white spot, and pink discoloration of the body. There is no specific treatment to WSSV, therefore pond sanitation and environmental control need to be practiced. This is objective, where the first is to observe the presence of white spot syndrome virus in fish sample by using WSSV rapid test kit. Next is to be able in handling the test kit in correct way by following the given procedures. Last objective is to determine the importance of WSSV kit in monitoring the health status of shrimp populations.
Material that we use for this experiment are WSSV test kit, petri dish, disposable grinder, microcentrifuge, scissor, and dropper.
We move for the material and methodology. First, a piece of hepatopancreatic shrimp tissue was cutting properly by using a scissor. The second, the sample was transferred into microcentrifuge tube. And then, the tube was added 10 drops of buffer solution, approximately 0.5 milliliter. Then, the sample was grinded properly into small pieces with the disposable pestle and let the particle to settle down. By using the disposable dropper, the sample solution was collected from upper layer and slowly apply 5 drops of sample solution into the application chamber well of the test kit. For the last, read the test kit result within 50 minutes.
Now, I will be continuing with result of WSSV. During the test, we used the hepatopancreas of the shrimp, and the result we got was negative. Based on the manual, when the line appeared on the control line C, means that the test was negative.
Hi, my name is Mr. Binti Abdulaziz, S64348. So today we will discuss about the results that we gained from the test kit that we did, which is white spot syndrome virus, and it turned out to be negative. Even though we follow all the procedure, but there might be a few factors that might lead to the negative results. First factors that might be the causes of our negative results is it might not have been the right time when we collected the samples, which means that the virus is not active yet. And the second is the level of the virus is too low that the sample test kit cannot detect it. Shrimp might have the virus before. They build strong immunity towards this virus.
So some test kits may be designed to detect specific variants, so understanding the prevalence strain in a region is important for selecting the most suitable test. And other than that, we must identify and assess potential risk factors that may contribute to WSSV outbreaks. This could include transportation of shrimp, introduction of new stock, or environmental changes. In this experiment, the result is negative, so it could be the risk on shrimp before test is reduced. As we did the test to ensure either the health of shrimp was having white spot syndrome virus or not, we realized that we need to emphasize the importance of public awareness and education in the shrimp farming industry regarding white spot syndrome virus.
We're moving to the conclusion.
As a conclusion, white spot syndrome virus (WSSV) is a highly contagious and devastating pathogen affecting shrimp population worldwide. In this lab, the use of diagnostic kits for detecting WSSV in shrimp sample has proven to be a valuable and efficient tool. These kits typically employ molecular technique such as PCR to identify the presence of WSSV DNA in shrimp tissue. The use of such kits enable rapid and accurate diagnosis in aquaculture setting to control the spread of the virus. Additionally, the application of this diagnosis tool contributes to the overall management and sustainability of shrimp farming by aiding in the early detection and prevention of WSSV outbreak. That's all from us. Thank you.
Hello, we are from Group 3 and this lab is about gram staining. Introduction to this lab, a gram stain is an examination applied to assist in determining bacteria and body fluids that do not ordinarily contain germs such as blood, urine or cerebrospinal fluid can be used to collect the tested sample. The sample can also be collected from the suspected infection site, such as the throat, lungs, genitals or skin, and bacteria are classed as gram positive or gram negative based on how they respond to the gram stain. The gram stain is purple in color, and when mixed with bacteria in its sample, the stain will either remain purple within the bacterium, gram positive, or change pink, gram negative, according to YAS 2022. The objective of this lab is to identify gram classification of Staphylococcus aureus and E. coli.
For methods of gram staining, first we need to label the slide using a pencil. And then we drew a circle with 1 cm diameter on the slide. And then we flame again to sterilize. A drop of sterile water is added to ensure the film appears on the loop. We sterilize again the metal inoculation loop to sterilize. A few drops of crystal violet added on the slide and let it sit for 1 minute. A few drops of grams iodine was added on the slide. A few drops of decolorizer added on the slide and let it sit for 1 minute. For counter staining, safranin was added, let it stand for 1 minute, and then we rinse it completely.
After doing this experiment, Staphylococcus aureus is showing blue color stain, which means that it is gram-positive bacteria. Meanwhile, Escherichia coli is showing pink color stain, and it is gram-negative bacteria.
Moving to the discussion, we can clearly see that Staphylococcus aureus appear in spherical shape. For the gram staining process, we can conclude that Staphylococcus aureus is a gram-positive bacteria, meaning it retains the violet stains in the gram staining process. Gram-positive bacteria have a thick peptidoglycan layers in their cell walls, which retains the crystal violet stain. For the disease, according to Matuzespa et al. 2023, the Staphylococcus aureus in fish can cause food poisoning because the ingestions of staphylococcal enterotoxin that produced by Staphylococcus aureus. Besides that, as stated by Bakers et al. 1985, Staphylococcus aureus is able to produce enterotoxin in shrimp, but its production depends upon a number of factors, including the relationship between Staphylococcus aureus and competitive microorganisms.
Next is E. coli. Based on the lab result, the E. coli shape appears to have a rod shape. E. coli are gram-negative, meaning that they do not retain crystal violet on the gram staining process. Gram-negative bacteria have thin peptidoglycan cell wall, which are the outer membrane contain lipopolysaccharide that does not retain crystal violet when immersed with alcohol. E. coli is a type of bacteria that commonly found in environment, food, and intestine of people and animals. E. coli infection can be transmitted through contaminated food, water, or through contact of animal or person.
In conclusion, we were able to identify the Staphylococcus aureus and Escherichia coli, which show a different characteristic. From our observation, Staphylococcus aureus exhibit a spherical shape through a gram staining. It is classified as gram-positive due to its ability to retain the violet stain. This gram-positive nature was attributed to the presence of a thick peptidoglycan layer in its cell wall. Next, Escherichia coli was a gram-negative and a root-shaped bacterium. In the gram staining process, Escherichia coli will stain pink. This characteristic is due to its thinner peptidoglycan layer in the cell wall, which does not retain the crystal violet stain. Fundamentally, this research not only illuminates the morphological and staining trait of these two bacteria, but also underscores the possible consequences of its existence in the aquatic environment. So that's all from our group. Thank you.